Hey everyone, and welcome to This Explainer. Today, we're going to completely transform your exam strategy. We're breaking down the exact silent mistakes that are literally costing students top marks in Cambridge 9700 biology. Seriously, pay close attention, because like 9 out of 10 students make these specific errors, and they just quietly drain away your grade. So we're going to expose these traps right now, before you ever step foot in the exam hall. Let's get into it.
Okay, so let's start with the classic cold open mistake. Take a look at this highly confident, but, well, completely flawed student answer. The one with the big red X. It totally reads like something straight out of a textbook, right? But it scores an absolute 0. Why? Well, it's way too vague, it's completely missing units, and it relies on these really generic definitions. But then, contrast that with the top band answer right next to it. That one is deeply contextual, it's entirely evidence-based, and it nails that precise, mark scheme-friendly terminology. Once you see exactly where that error creeps in, honestly, doing it the right way becomes almost second nature.
And here is where so many people trip up. 9 out of 10 students fall into this exact cell structure trap. You know, they get faced with a beautiful, complex scenario like this plant and animal cell, and they just kind of panic. They default to memorize generic terminology. They totally fail to link what they know directly to the specific scenario provided. So, for instance, they'll just write down a textbook definition of a mitochondrion, but they completely forget to link its actual function to the specific biological stimulus the examiner gave them. That is exactly how you lose easy marks.
All right, moving on to Section 1, the high mark framework. We're talking deconstruct, solve, and evaluate. Let's break it down. Look, examiners don't just want a list of facts. They actually want to see you solve a problem. So mastering these 4 specific steps is absolutely crucial whenever you're staring down a tough question. First up, concept identification. You've got to pinpoint the core biological principle. Step 2 is evidence and data integration. You need to extract the specific numbers, the trends, the raw data straight from the prompt. Step 3, scientific reasoning. This is where the magic happens. You connect that evidence back to the concept using logical if-then statements. And finally, step 4, examiner wording. You've got to refine that final answer using the exact, precise terminology they're looking for in the mark schemes. Let's actually look at how this plays out.
So here's that framework in action, specifically looking at phloem sieve tube elements and companion cells. Notice how a top-tier candidate doesn't just, generically describe the phloem. No, they pull specific values straight from the data table to really anchor their answer. By systematically extracting that info and applying the correct biological principles, like the active loading of sucrose, they completely move past superficial answers. They show that connected, conceptual understanding that examiners absolutely love to reward. This is how you guarantee you're operating in that top-tier A cognitive domain.
Let's apply this to the mammalian heart and really see how to level up your answers. A lot of students just get stuck at the bottom, descriptive level. They basically just answer, what is it, and maybe label some basic external structures. But the high-performing candidates, they push further. They move up to the analytical level, answering, how does it work, by explaining things like the maintenance of pressure. And then they hit that top-scoring evaluative level. This is where you're actually questioning assumptions, evaluating the validity of the given evidence, and fully justifying your decisions. It's a total game changer.
Next up, Section 2. Mastering technical methodology and statistical tools. Okay, before we dive into the numbers, I want to share a massive secret straight from the examiner reports regarding practical work and data interpretation. Even if your final numerical calculation is completely 100% wrong, you can still score highly. It's called the method mark strategy. To make this super tangible, just memorize this simple table. When you're hit with data, you need to know your tools instantly. Let's break it down. If you need to compare the means of 2 sets of data, boom, student's t-test. If you're analyzing categorical data frequencies, that's a chi-squared test. And if you're trying to figure out the relationship between 2 continuous variables, you're going to use a correlation test like Pearson's. Knowing this right off the bat will literally stop you from freezing up during the exam.
Take a really close look at the flowchart on this screen. When you're evaluating limiting factors, things like light intensity, CO2 concentration, or temperature, your very first step is to deconstruct the command word. Is the examiner asking you to explain, or are they asking you to describe? Once you figure that out, you have to explicitly link your data to the core concept using causal relationships. And here is a huge examiner alert: cause versus effect confusion. Candidates mess this up all the time. They fail to distinguish between the actual cause of a biological phenomenon and its effect on a graph. You absolutely must show clear, step-by-step working and logical reasoning to snatch up those method marks.
Moving right along into Section 3, navigating the top 3 examiner alerts.
All right, straight from the Cambridge Principal Examiner reports, here are the top 3 highly specific traps, absolute mark killers, that you have to avoid if you want that A grade. Trap number 1, terminology and unit neglect. Number 2, the generic answer trap. And number 3, cause versus effect confusion. Think about it. If you calculate a rate of flow but completely forget to write the units, boom, you lose the mark. If you accidentally reverse the logical sequence of hydrostatic pressure and mass flow, you lose the mark. Or if you write a flawlessly memorized textbook definition, but it has literally nothing to do with the specific scenario provided, that is an instant red flag for the examiner.
To really drive this home, I want you to zoom in on the center of this nephron slide, right at the calculation section with the red examiner alert. Forgetting one simple unit here completely throws off the entire answer. Like, the student did all the math perfectly, but without the units, that numerical value is basically biologically meaningless as far as the mark scheme is concerned. Precision is absolutely everything.
And this leads us perfectly into a concept you really need to wrap your head around: contextual misalignment. To put it simply, contextual misalignment happens when you write down every single thing you've memorized about a topic, but absolutely none of it actually answers the specific scenario sitting right in front of you. Sure, you're providing correct biological facts, but they're the entirely wrong facts for that exact moment.
This slide on the negative feedback control of blood glucose is literally the perfect case study for this. Look at how the flawed answer just vaguely states, hormones change sugar levels. That's the vague trap in action. But check out the improved, top-band answer. The way high-mark candidates secure the bag here is by referencing cross-topic connections. They don't just ramble about insulin. They link it directly to cell signaling and explain how glucose transport proteins are actually moving within the cell. They're connecting the dots across the entire syllabus. That specific level of analytical depth is exactly what the examiners are hunting for.
All right, Section 4, Final Exam Takeaways and the 10-second challenge. Let's see what you've learned.
So we've gone through the most common traps, and we've built up your completely proven 4-step framework. Now it's time to put your new examiner-level vision to the test. I'm going to give you 10 seconds. I want you to look really closely at the methodology and the examiner verdicts coming up. And your goal is to pinpoint exactly why the student lost their marks. Are you ready? Go!
All right, time is up. Did you catch it? The big reveal is right there in the Phase 3 examiner's verdict section at the bottom of the slide. The student completely failed to include proper units in their calculations. But even worse, they didn't match their terminology to the specific unfamiliar context the question provided. They clearly memorized exactly how an enzyme-substrate complex works, but the second the exam threw a new real-world biological scenario at them, they just panicked and regurgitated a generic textbook answer. They completely missed the context-specific application.
Listen, if you take absolutely nothing else away from this explainer, please remember the method mark strategy. Always, always, always show your clear sequential working. Marks are awarded for the actual process of logical reasoning. If you can identify the core concept, extract the right data, and clearly apply your scientific reasoning, you're actively securing marks at every single step, even if you make some silly, minor numerical error right at the very end. Let me leave you with this exact quote from the Principal Examiner reports. Top-performing students exhibit connected, conceptual understanding transferable to unfamiliar contexts. So I want you to ask yourself right now, if Cambridge throws a wild biological scenario at you tomorrow that you've never, ever seen before, will your knowledge break down, or will you use this framework to break it down? Master the method, guys. Thanks so much for joining me for this explainer, and as always, keep studying smart.